Well, in the last lecture about DNA physical structure, we talked about the fact that Levine's model of a tetranucleotide system had been shown to be incorrect with Chargaff's data. Also, within Chargaff's data, we learned that there were various rules about A was linked to T by 2 hydrogen bonds, G to C by 3 hydrogen bonds, and that there was other components about double-stranded versus single-stranded DNA, some basic principles. So now that the tetrahedral hypothesis is gone, what do we have? Well, we ended up with the last discussion in that lecture, whether it was about Linus Pauling. Linus Pauling proposed a 3-stranded model that had the phosphate groups actually on the inside. So it was this 3-strand model, and that was kind of, I mean, he was a very prolific individual, highly respected scientist, Nobel laureate. So, you know, it seemed like that might be a good model. But there were some people working on it who eventually came up with an alternative that turns out to be the primary model that as we understand it today. So let me share this with this. So this part of the lecture is really about taking all the pieces that we have and putting those pieces together, okay? So let's look at the people that did that. Most of us know, I mean, again, almost everything in this first module of genetics is remedial, and we expect it to be remedial, but we we all know that Watson and Crick received a Nobel Prize, and along with some others, for developing this final model structure of DNA. And they literally sat around with some, you know, pipe things you would use in a chemistry lab to hold pipes up to hold in the heat, and some other structure, and physically made some models. This is the actual model. You can actually see it in a museum where they work. And the 2 individuals involved were Francis Crick. He was a graduate student at the time. He was working in Cambridge in the UK. And interestingly, he was working on X-ray diffraction. Now, he was not working on DNA at the time. He was working with polypeptides and proteins, trying to understand the structure of proteins. But what was important that he was working with X-ray diffraction. X-ray diffraction, of course, we'll talk about in a moment, but I was using X-rays to blast it through compounds and see how the pattern that showed up. The other individual, a very aggressive young man, was James Watson, was an American. He had gone over on a postdoc to Cambridge, and he set out to figure out the structure of DNA. That was his goal in life, was to figure it out. And he and Crick became collaborators, and they started working on this project. Now, they sat down and they thought about, okay, what all do we know, right? What things do we know? Well, we certainly know that under the basic models that we would have, that A and T somehow have to link together, and G and C somehow have to link together. Now, they had actually talked to Chargaff, okay, and they knew from Chargaff that A and T were going to link up with double bonds, so you're going to have some double bonds here. And I better get a color that we can actually see, maybe red, there we go. And so you'd have a double bonds going across here. These were hydrogen bonds. They were known to be hydrogen bonds. AT would have another hydrogen bond going across here, holding these together. And then the GCs were 3 bonded units, okay? So they have bonds that look something like this. So we would have this, and so they started looking at these components and said, all right, this is what we know. And they started building these models based on this. They knew that base pairing, which had come from Chargaff, had to work, that T and A were hooked together, and they were chemically, at least the predominant form in DNA involves 2 hydrogen bonds, and GC involves 3 hydrogen bonds. The problem they ran into when they did this was their model wasn't nice and straight. Okay, their model didn't look, you know, even early on, it didn't have a nice dial pattern like this. Why? Because these hydrogen bonds that are down here on GC would pull the system in tighter, and the ATs would have be a little less tight because there's only 2 bonds. And so instead of getting a sort of a parallel structure like we might see here, you ended up with sort of a structure that had sort of an irregular pattern, and the irregular pattern would be based on where the T's were and where the G's were and the C's were in the sequence, right? So they felt, okay, this doesn't sound like this is the correct way to work, but we're going to work with it. So they kept looking at it, looking at it, looking at it. Now, very importantly, at the same time, there was sort of some really clear evidence that they were correct, that this idea that there's this bumpy model was supposed to happen was wrong. And those data came from Rosalind Franklin. She was working at King's College, Cambridge, and she was also doing X-ray diffraction, but she was doing it on DNA. And one of the things that was really important is that she could extract really good, high quality DNA. That was something that Watson and Crick themselves had not been able to do. And it was actually very difficult, Nowadays, we do it with just a simple kit. You can do it in, we teach it sometimes in high school or grade school, where you just take a jar and throw some hot water in it and a little bit of detergent and some tissue that you want to, digest, some wheat or something, and shake it around a little bit, pour a little alcohol in after a while, and you get a really beautiful structure of DNA, pile of DNA. And of course, not very clean, but you can clean it up. Well, they couldn't even, the techniques there were not that advanced at the time. Now, of course, we have little tubes and things. We can isolate DNA within minutes, robotically, and all sorts of other ways, and very, very clean. But she could get good, clean DNA. And she was working with Maurice Wilkins, and that was a collaborator, and they were working on this X-ray diffraction. And as I said, we talked a little bit about X-ray diffraction. Basically, we just take an X-ray source, run it through a shield, which would then form it into a beam. The reason it's a beam is it only goes through certain parts of it, and it's shielded. And that beam then can go through and would beam through whatever system you were looking at, okay? Whether it was proteins, as you might be for Crick, or in case of Franklin, it would be DNA. And then that would give some type of pattern, and it would give an X-ray diffraction pattern. And this is the pattern that was seen. Now, there's a lot of controversy about how Watson and found out about this. Okay, some say that it was provided to him by Franklin. Some say that he snuck into the lab and did it. It's one of those things that pops up in the history of systems. But anyway, one way or the other, he became aware of this pattern. Now you look at that pattern, you say, it doesn't look like anything that would be important to me, but it is. Okay, so for one thing, it's like going under a building. I picked the Eiffel Tower here just because it's pretty, and you can stand under it. If you walk under this structure and stand under it and look straight up, okay, this really what you're looking at here. You're kind of looking at the DNA from on the end, right? And what they were seeing is that there were these two basic patterns, okay? There were these outlines on the outside that looked something like this, these dark areas, and then there were these regular steps on the inside, okay? Well, immediately what you know is that the Linus-Pauling model is wrong, okay? If the Linus-Pauling model was correct, there should be 3 of these dark staining areas, somehow maybe separated around, one over here, and one over here, and one here, so they were equally dispersed around. But there's only 2. So this told them that they were correct, that Pauling was wrong, that DNA was most likely double-stranded. Okay, and then these center parts right here, they turned out to be the nucleotides. So unlike Pauling's model, which had the phosphate groups on the inside, this suggested that the nucleotides were there, and they were in very regular step pattern as they walked through. Okay, so you had this nice little pattern that worked through it, right? And it's just like, again, looking through the Eiffel Tower or something. Here instead of the 4 bases, this would be what a tetrahedral model would look like if you were to do it with an X-ray diffraction. It would look like this. Well, this is not a tetrahedral model. Now, there's a couple other things. One, it's very regular, right? Notice that the distance here, there's not a lot of fuzz way up here and then fuzz off over here. It's very regular in distance between here. So not only are the steps very regular, but the outside distance seems to be fairly regular, suggesting that there's a sort of a constant through it. Okay? Turns out that distance is approximately 2 nanometers. It's at least at the time, that was the best estimate of the distance that that it had. Okay, and these bases in here, these steps, were about 0.34 nanometers. Those are important numbers. Okay, they're part of the Watson-Crick definition, and they're important because we will often ask you, okay, if you've got a piece of DNA that's double-stranded and it's 1,000 base pairs long, or how long physically long is that? We multiply 1,000 by 0.34 nanometers or something. Okay. All right. So this is all the information. This is the physical evidence. This is the blueprint. Okay. This is what they have. This is what Watson sees. This is what they talk about, and they come back and they say, okay, then this is it's just putting all of it together, the pieces. You say, all right, what have we got? Well, we've got these bases. We know the chemistry. We know the model from Levine. Levine was correct about it being made up of nucleotides. He was incorrect about how those nucleotides connected. Chargaff then came along and said, yeah, this is, there's a very specific model of how these connect together. Now we've got a physical blueprint, and somehow we've got to make that into this helical form of DNA. And again, notice it's very regular in distance here. So somehow the sides of it have to be regular. And once again, if we go and we look at what Watson and Crick were doing, they were sitting there and they were saying, okay, I've got this model. I'm going to take these phosphodiester bonds. I'm going to hook them together. And then, of course, I'm going to take my bases and I'm going to have to set them up some way like this, okay? This is my blueprint. Now, once again, if we do that and we start making these connections, we've got 2 bonds here, okay? And we've got 3 bonds that come up down here, okay, between T and G. And again, we've talked about thousands and thousands and thousands of these bonds, right? Because you've got thousands and thousands of base pairs, or even a tiny piece of DNA. So they're looking at this, and again, what it's doing is it's not a nice, pretty model like you see over here, okay? What you're ending up with is some kind of model that every time they would put it together on their little stand would zig and zag kind of in and out like this, right? And sometimes it'd be close in the center, sometimes it'd be out. And they said, well, that's clearly not what this model over here says. This model says there's a consistent distance in between here. All right, so they started working, thinking, and Watson goes off and they think about this for quite a while. And then they said, well, how do we, if we've got this problem, we're trying to fit it within these diameters, what do we do? Watson, literally in a bar one day, comes running back, and it's one of those eureka moments, right? Eureka, I found it, I think, right? Not what he said, but he said, oh, what we need to do is flip it over, okay? If we turn it over, then we've got nice, simple bonding structures here that will work. Sure enough, they tore their model apart, at least one side of it, started putting it back together, and lo and behold, you see the dynamics of it work out. You now get, if you just have anti-parallel, okay, so remember these are anti-parallel, so we're going 3' to 5' on one side, but opposite of that is 3' to 5', but going in the other direction on this side over here, okay? So we end up with this model that looks like this. Well, everything fit now, okay? The base pairs were the proper distance apart. You didn't have the bases going on and then sometimes folding down because of the extra angles. You ended up with this very regular step pattern of about 3.4 nanometers. That's approximate, and about 2 nanometer wide. Okay, and those are good enough numbers for government work. Okay, as we say, you can use those in your calculations. So this is it. It said, here's what DNA looks like. Okay, and they proposed this model in the 1950s, and in doing so, they said, okay, here's the model. They wrote up a short little paper. It's about 1.5 pages, put it in the journal Nature. People looked at it. They said, oh, wow, that's important. It wasn't until 1962 that they received the Nobel Prize for it. And by that time, Rosalind Franklin, who'd been exposing herself to all these x-rays, had actually passed away. So she did not receive the Nobel Prize for it, but that was it. But they wrote it up, but in their writing, they not only proposed, okay, here's the model of how it looks, okay, remember, and this is remember all the things that DNA has to be able to do, right? Or at least any genetic material has to store lots of information, has to be easily replicated, has to be replicated with high fidelity, and all that. Well, they proposed a model of replication at the same time, okay? They proposed this model where we're just gonna break these little hydrogen bonds, we're going to use one side as a template for a new strand and the other side as a template for another new strand. And we're not going to break them apart, replicate them, and then put them all back together. We're actually going to use this side as one model, and we'll make an anti-parallel strand to it. And then we'll take this side over here and make another, and make an anti-parallel model for it. They proposed that same thing in the same paper, and that's one of the things we'll talk about when we get to replication. What took so long for some of this to work out was trying to put all these pieces and finding scientific evidence that the models were correct, trying to refute the alternatives. But anyway, they had said this is what it looks like. Tetrahedral model was wrong. Linus Pauling's model was wrong. We've got our phosphate groups on the outside. We've got our nitrogen spaces on the inside. We've got these anti-parallel strands.